This video is about updates on parasites of the immunocompromised host, particularly newer therapeutic updates on Cryptosporidium, as well as clinical pathologic updates about Toxoplasmosis. Let's talk about Cryptosporidium first. I have an older lecture on Cryptosporidium. Please watch that video if you haven't already. Link is in the description below. But in a nutshell, Cryptosporidium is a parasitic coccidian protozoan, which mainly infects the gastrointestinal tract and causes intractable watery diarrhea. We usually get it by ingestion of water from contaminated drinking supplies or even from water parks or swimming pools. And immunocompromised people, especially HIV/AIDS patients, tend to have more severe diseases caused by this protozoan parasite. So let's talk about what's new on treating cryptosporidiosis. Last time we checked, in immune-competent people, nitazoxanide has found success in cryptosporidiosis treatment. However, the same drug has a poor track record in treating those with HIV/AIDS. Other drugs, such as azithromycin, still had questionable efficacy. There's this 2022 published study in the Indian Society for Parasitology by Dal et al., which, apart from summarizing the biology, the life cycle, and pathogenesis of the parasite, they also discussed the development of anti-cryptosporidium drugs currently on the pipeline. The link to the full study is in the description below. Based on the review article, there's quite a list of repurposed and newer drug candidates undergoing preclinical trials to try to combat cryptosporidium infection, both in immune-competent and immunocompromised patients. But seemingly effective candidates are only effective in silico, in vitro, or in animal studies, and would warrant more studies, especially human clinical trials. Examples of these drugs include paromomycin, the antilipidemic drug atorvastatin, and even one repurposed anti-malarial drug. Now let's talk about Toxoplasma. I also have an older lecture on this parasite as part of my coccidian lecture series. Go watch that video if you haven't already. Link is in the description below. As a quick recap, toxoplasmosis is caused by the parasitic protozoan Toxoplasma gondii, also considered a coccidian parasite. It causes a lot of clinical diseases ranging from mild fever and lymphadenopathy to chorioretinitis, even preterm labor or congenital problems, and even encephalopathy. It causes these varied diseases due to its capability to hitch a ride inside macrophages and then navigate and then spread throughout the entire body, infecting most tissues. This is most true especially in patients who are immunocompromised, including patients with full-blown AIDS. Due to its worldwide prevalence levels, as well as the number of diseases associated with it, there have been quite a number of studies regarding Toxoplasma and Toxoplasmosis. We will discuss two relatively recent articles about this notoriously ubiquitous parasite. There's this study by Babekir et al. In 2022 which looked at the relationship between Toxoplasma gondii serum levels and liver biomarkers. Now, I'm not really a fan of the MDPI line of journals, and I have some reservations regarding their results and conclusions, particularly due to inconsistent results depending on how they modeled their regression equations, but we can still learn from their general findings. Now, if you want to take a look at the full article yourselves, the link is in the description below. After controlling for potential confounders like viral hepatitis, alcohol intake, and even potential environmental exposures, their study concluded that there is a significant association between a positive Toxoplasma IgG antibody and serum biomarkers for liver injury, such as ALT, AST, GGT, and ALP, and even an overall increased risk of chronic liver disease and non-alcoholic fatty liver disease. They recommend that their findings may help support the use of liver biomarkers together with a non-radiologic index to assess liver complications from toxoplasmosis, especially in areas where radiologic testing is not routinely available. They also mentioned that high exposure occupations such as construction increase the odds of liver injury associated with a positive Toxoplasma antibody. Now again, I invite everyone to read the article to look at the limitations from their results. The last article is definitely the most interesting one for me. Let's look at an article by Taghipour et al. Last 2021 in the Journal of Parasitology Research. Now in their study, they did an in silico or computer-aided methodology to look for potential targets in the development of Toxoplasma gondii vaccine. Their research focused on the Toxoplasma calcium-dependent protein kinases or CDPKs, which are important in a lot of Toxoplasma functions like motility and cell infection, even cell division and cell growth. Now, CDPKs are important in the life cycle and pathogenesis of Toxoplasma gondii, hence targeting these by a vaccine would prove to be clinically beneficial, at least theoretically. Their study also used an artificial intelligence-based approach to predict which of the identified epitopes have the most potential to be immunogenic and non-allergenic, which are two characteristics of good vaccine candidates. Now, this article resonated with me since back in my undergraduate days, the selection, the isolation, and even the creation of possible vaccine epitopes were very much done manually, requiring tons of samples and funding, months upon years of isolation of stable proteins, and definitely infinite patience. But with the advent of advanced AI technology, vaccine research has really gone a long way. I'm very much optimistic that vaccine research will be much more efficient and that we will get to have at least some potential vaccine candidates for a lot of important parasites in the near future.